right, we are here to talk about regulation of cell signaling. We're also going to talk about disruption. So what happens when you have disruption in a cell signaling pathway? And again, when you think about cell signaling, go ahead in your mind, be thinking reception, transduction, response. So what happens if that gets disrupted? And then why and how do we regulate? Why do we need to regulate that? And then how do we regulate it? So the first is I can explain how a cell signaling pathway can be disrupted. Well, let's look really quick. Hopefully this will work. I was being a little cranky yesterday, but yep, perfect. Let's look at a cell signaling pathway and talk about maybe some areas or places that we could see. My messy desk, sorry. Some disruption. All right, so here we have a receptor, which has a ligand bound to it. So it's got reception. The intracellular part of that receptor has been activated. We go through transduction, which is generally some kind of chain reaction, maybe a phosphorylation cascade or some other kind of chain reaction that leads us to a response. So these molecules all get activated, and then this last one will activate this response. And that response can be turning on a gene for transcription. It could be going, telling the cell to go through mitosis or apoptosis. It could be telling the cell to release a neurotransmitter. I mean, there's all kinds of different responses. So what happens if a cell signaling gets disrupted, or how could that happen? Well, there's a couple of different things. You could have an antibody that comes in and binds to the receptor, and what that can do, it can do one of two things. It can either block the receptor so the ligand cannot bind, so that would prevent reception, and that means this receptor would not be turned on, so you wouldn't see these little activated lights, whatever they're called. Transduction wouldn't occur and response wouldn't occur. So you basically would be shutting down that cell signaling pathway. Or sometimes what we see this with Graves' disease, is the antibody binds to the receptor, but it pretends like it's the ligand, and that turns the receptor on and it keeps it on. So that's a disruption as well, because we need these to only happen when we want them to happen. And if they're happening 100% of the time, they're always on, that's not necessarily a good thing either. And so we see that sometimes too. That antibody binds to the receptor and it leaves it in that active state. And so we have transduction in that response all the time, even when we don't need it. So that's one place or one way that we might see disruption, is at the very beginning. We might also see some kind of mutation in this receptor. And when that receptor is mutated, it may cause it to stay on all the time, or it may cause it to not do its job when the ligand binds. So it may make it dysfunctional. It may make it non-functional. So dysfunctional would mean it does the wrong job. It could maybe start a different transduction pathway and a different response. Or non-functional means it doesn't get a response when it should. So even when that ligand binds, there's no response. So those are some different ways the receptor could have a disruption that affects the pathway. Then maybe everything's working fine up here, but maybe one of these molecules is mutated or has a problem. And so maybe this one's the problem, and it doesn't do its job. That means none of this would happen. So basically you're stopping your pathway right there. You don't get your response. Same thing if one of these is mutated. Or maybe the ligand's not bound. So maybe the receptor is turned off, okay? But this guy is mutated to the point that it's activated. So even though it's not getting the response from the receptor to be active, it's activated. And so it makes this response happen even when it's not supposed to, even when there's no ligand bound. So you could see that happening. You also may have other chemicals out here that come in and impact that pathway, either causing it to happen when it shouldn't or turning it off when it needs to happen. And so you could have a problem in transduction or disruption in transduction that means that your pathway either is overactivated, or and you get too much response, or it's inactivated and you don't get any response or not enough response. And then, of course, you could have a disruption in the response itself. So it could be that reception and transduction are happening just fine, but maybe the transcription factor, whatever reason, is mutated and it can't bind to the DNA, so it can't turn the gene on. Or maybe one of the Cas bases in the apoptosis pathway is mutated, and so it doesn't cause apoptosis. And so those are some disruptions you might see down here at the bottom in that response portion that allow the response to either happen too much or not enough. So sometimes there is a disruption. It could be a mutation in the receptor or some other component along the pathway that could increase or decrease activity. Again, you could have interference by another chemical, and that could cause disruptions as well. So what we generally see when a pathway is disrupted, we either see too much response or possibly too little response, but oftentimes what we see is no response. So it's either overactivated or it's completely turned off. And that can be a problem, obviously, if that's a response that we need. We talked a little bit about endocrine disruptors, and so endocrine disruptors are just chemicals in the environment that alter the pathway of these signal transduction pathways in the endocrine system. And we see this a lot. We think this is probably what's going on with a lot of people who are having fertility issues. A lot of cancers, we're seeing cancers on the rise, compared to 100 years ago. And we think that's probably some things in the environment, and some of those are disrupting these pathways and causing mitosis to happen when it shouldn't, or are causing things to not happen when they should, and it's creating problems all over the place. All right, so those are some, explain how self-signaling pathway may be disrupted and give examples of what happens when it's disrupted, so overaction or total inaction. I can explain how the same ligand can give a different response. Okay, so let's say we have this ligand and it binds to this receptor, and let's say this is the alpha receptor, and it gives this response. Well, maybe on a different cell there is, I'm trying to find my pen that I just had. Maybe on a different cell, there it is. There, it's right beside me. It's too close, that's the problem. On a different cell, there's a different receptor, and when that same ligand binds, okay, so maybe this is adrenaline, but because maybe this is a liver cell, it's going to cause sugar to be released, glucose to be released. If this is a intestinal cell, then it's going to have a different transduction. So this might be a beta receptor, and it's going to have a different transduction pathway, which is going to give a different response. And so instead of releasing glucose, your intestinal cells are going to slow down. So they're going to slow down digestion because you got more important things to worry about. And so that's where you might see the same ligand. I need to zoom out a little bit on the same ligand giving you two different responses, because these different cells have different proteins in them, and so they're going to have different transduction pathways, or in this case, different receptors. Then let me see what else. Okay, the next one is I can explain the importance of feedback loops and cell signaling pathway. And we kind of talked about that a little bit in the apoptosis video, but basically, and a little bit with disruptions a minute ago, when we signal for these pathways to happen, it's because we need them to happen. Okay, so if we signal, maybe this is a neuron and something binds and we go through the action potential to release serotonin or dopamine or whatever, there's a reason we need that. We need that response. Or maybe we need to do mitosis because I got a cut, a paper cut, and I need to do mitosis to heal. There's a reason for that. We want to make sure that only happens when it needs to, because we don't want to waste energy. Organisms that waste energy don't tend to survive. And when you do these pathways and they're not necessary, then you're wasting energy. So we don't want to do that. So we want to be able to turn them off when we need them off. But again, when we need that response, we need that response. And so we want to be able to turn them on when we need to turn them on. And so it is going to allow us to maintain homeostasis, to use these feedbacks only as we need to. It's going to allow us to respond to the environment. If you remember, those are two characteristics of life. And so if we can't do these when we need to, we might not be able to respond to the environment. So everything you do, or most everything you do, is the response of a self-signaling pathway. So if you're shivering because you're cold, if you're sweating because you're hot, all of those are signaling pathways. And when those get out of control, that can be dangerous. And again, it could be dangerous. It could also just be wasteful. All right, so some examples of feedback. Looking at the PowerPoints, I know if you did the Nearpod, good gracious, what's going on? If you did the Nearpod, you saw this. Let's see, let me find the right, there we go, there's an example. Here we have a G protein coupled receptor, and we have this pathway in which the receptor activates these molecules, and it's like a chain reaction. So RAS activates RAF, RAF activates ERK, that's kind of fun to say, and then ERK goes in and does transcription, so that's the response. In a normal cell, what happens is when we have plenty of ERK, it will go up here and stop the RAS. It'll turn it off, because what it's saying is if you have plenty of ERK, we don't need to be making more ERK, okay, because we got plenty. I feel like I'm speaking a different language. So what it does is it turns off RAS so that RAS doesn't make RAF and RAF doesn't make ERK, and so it kind of slows things down so that we get a normal transcription. But what happens when it's a cancerous cell is a mutated RAS stays on, so even when ERK goes back up here and says, hey, you need to cut off, the mutant rat says, RAS says, talk to the hand. I don't really care what you think. I'm going to keep going. And so you're going to keep making RAF and keep making ERK, and you're going to keep doing proliferation. And so you see this, this lack of control that leads to cancer. And that's usually what we see with negative feedback, is something downstream goes back and turns off something further upstream. And that just, again, saves energy. It allows us to only get this response when we need it. So that is one example of feedback. We could also have positive feedback. We don't see that a whole lot with cell signaling pathways, but we could see that happen where we need a certain response to occur, and so we keep sending responses and we multiply and we amplify those responses until you get that final response, and then you shut it down. And so those are some examples of feedback. So why is it important? It's important to regulate because we need that response when we need it. Okay, otherwise it could lead to disease or death. But we don't want to do that response if we don't need to, because that could be dangerous or at the very least wasteful as far as energy goes. So that is why we want to be able to regulate that, and we use feedback loops to do that. And so here's an example of a negative feedback loop. There's tons and tons of examples, but what you're always going to see with negative is the response is going to turn off the process that causes that response. So ERK is one of the responses, is a part of the further down the pathway. It's going to turn off RAS, so we're not going to make any more ERK. Okay, so it basically stops the production of itself or activation of itself. Positive would be if ERK went up here and said, hey RAS, we need more ERK, and so it made it have maybe multiple pathways, and we got lots and lots and lots of ERK, which is what we see in cancer. So there are, again, times that we need that, but for the most part, we're going to see negative feedback with cell signaling loops. So if you have any questions about pathways being disrupted and regulation of pathways, please let me know. And that's it.